So in this video I'll be talking about materials and methods of this paper. Uh, when I found out there are three main techniques which they have used. One is microdialysis, another is the wavelet analysis in EEG, and third is optogenetics. Okay, so I'll be talking about two of these, microdialysis and wavelet analysis. Rest we will discuss later. So for wavelet analysis, first I'll go to MATLAB. Let us see what I have done here. So now EEG signal is a complex signal that is decomposed summed activity of multiple signals. So to understand wavelet analysis, let us start out with a simple signal. So I have a simple signal S. So let us try to plot it. Okay, so what we see here, it is a simple signal, a sinusoidal waveform of a particular frequency of a particular length or time. Okay, so if you do FFT of this signal, it will give a single peak at say 5 Hz frequency and no peak at other frequencies. And but the thing is, FFT is, it doesn't give you the time information. Wavelet, if you do wavelet also gives the time information. So what wavelet will give you, a single peak at 5 Hz at all time points. Okay, it also differentiates in the time domain, and no peak at all time points in all other frequencies. Okay, so let's try to do the wavelet analysis also. So this is the command for wavelet analysis. CWT is a function which stands for continuous wavelet transform. S is the signal. These are scales. They are like representation for frequency, and MORL stands for Morlet wavelet. Okay, so let us do it now. My C output variable has the wavelet output. Okay, so let's try to plot it. Now this is the plot. Okay, so in 3D you can see like it has given peak at one frequency. Now these are frequencies in this axis, these are time points. So at all time points, but only in one frequency, this is the 5 Hz frequency signal, no peak at other frequencies, and this axis is the power. So now if you convert it into a 2D image, okay, so this is how it gets converted into 2D. Okay, so now you can see at all time points, this x-axis is time point, y-axis is the frequencies, only at 5 Hz frequency. Now this 10, actually it's at 5. So only at 5 Hz there is a color change, otherwise it's all green, means no power. Okay, so this is how you can see they have done the wavelet analysis. So some power at the lower frequencies at all time points, but here you can see the red thing, it's only at certain frequency, at in certain frequency and at certain time point, not at other time points. So this is how they have done wavelet analysis. Let us see the microdialysis also. Now for that, let us see the setup for microdialysis. Now microdialysis is actually dialysis performed inside the brain with a very small probe of very small diameter. Okay, so this is the probe. So you can see it has a shaft, two outlet and inlet, and one semi-permeable membrane. So the concept is that artificial CSF, ACSF, is perfused inside this semi-permeable membrane, and outside this is the CSF. So because of concentration gradient, some solutes will pass down their concentration gradient. Okay, whether from the CSF to ACSF, if you want to study the neurotransmitters present, or if you make a high concentration of some drug in the ACSF, then you can actually, it's called reverse microdialysis. You can actually make the drug to, like if drug more concentration inside, you can make the drug to go out into the CSF. Okay, so it can be also a drug delivery system. So this is you can see microdialysis probe, inlet, outlet, and the probe is inside the guide cannula, which is fixed inside the brain. Okay, so I hope this kind of learning exercise helps. Let us see the response. Yeah, thank you.